Good evening, everyone. I hope you all are doing well. Today, we will continue from where we left off in the previous class. This will be the next part of the topic discussed earlier. So, without taking much of your time, I would like to invite Mr. Rupinder sir to continue today's session. Sir, the stage is yours. Thank you.
Okay, thank you, Priya. I'm just sharing the screen. I'm just wait. Is presentation visible to everyone? Please confirm. Hello.
Yes, sir.
Yes, sir.
Okay. So we left the topic list on the okay nomenclature, na? Okay. So any doubt, any query regarding any topic from the previous chapter which we have discussed yesterday? Anyone? Any student?
Sir, I have a question.
Yes, ask me.
Sir, if uh, in a 10 or if a 10 or 20-year-old case is reopened, then
Hmm.
can DNA fingerprinting still help in solving the case? If yes, then
Sure.
how much reliable will be it?
Actually, that is depend upon the condition of the sample which you have collected there, which we have in our custody. Okay, if the case is more than a decade or 2 decade, okay, means 10 or 20 years, then if it is reported today, okay, if that case is reported today, and we have to go there and collect the sample, so it will depend upon the condition of the sample which are present there. Okay, but accordingly in a DNA forensic after 20 or 10 years, okay, we will collect only bones. Okay, अगर कोई हमें केस मिलता है like if it is a bone of it is a case of murder, okay, then we will recover our bones like a femur, which whole body skeleton bones can be recovered. Okay, but if we want to extract a DNA from the tissue, then it will be difficult. It will not it will be impossible. Okay, because tissue will already decomposed and it will on the skeletal stage. Okay, after 10 to 20 years. Second condition is that when we have a sample in our custody, like in FSL, and there in some instances we have to need to reopen the case. Okay, by the permission of court or under any circumstances, then it will again also depend upon the condition of the sample present in the stored condition. Okay, so DNA can be extracted after 10 to 20 years. No, it's no doubt. इसमें कोई भी ऐसे doubt नहीं है. So it can be solved. Okay.
Okay, thank you, sir.
Hmm. Anyone else? Have you heard about the Nithari case? Nithari killing, Sector 36 movie देखी होगी किसी ने? Anyone? Nithari case, Ayushi Talwar case. I think Ayushi Talwar है ना? Ayushi Talwar case. And
Yes, sir. Yes, sir.
one more.
Yes, sir.
There was one more case. I did not remember now. It was a very good case on a DNA. Actually, it was a tandoor case, but उसमें क्या है ना? In that type of scenarios, there were not the time lapse between that 10 to 20 years. But the tissue of the we can say bodies of different children in Ithari case were decomposed at that time. Okay, and only hello. Okay. Okay, so the forensic scientist will cover only the bones, like the femur bone, short bone, long bone. Okay, so from that bone they recover the DNA and match with the reference sample, which were collected from their parents, their belongings. Okay, etc. So anyone else? Any query? Any doubt? Anyone else? Please reply with yes or no. Ria, please reply with yes or no if they are commenting in a comment box.
Everyone, please unmute your mic and say yes or no, sir.
No, sir.
No, sir.
No, sir.
No, sir.
No, sir.
Okay,
No, sir.
okay, good. Okay, good. Okay. So we have discussed about the nomenclature of DNA marker. Okay. So how we will nomenclature, how we give the naming to different type of marker like STR marker like TH01 is a type of marker. THs come from the tyrosine hydroxylase and amino acid, and 01 is the number of, we can say, repeat region present in the DNA. Okay, so there are different type of condition in which we have discussed about the nomenclature of the DNA. So there is some example. One example is that D16539. So D represents the DNA chromosome number 16, as is the single copy of sequence, and 539 locus described on the chromosome number 16. Okay, so this is the total, we can say, set of chromosome, and along with the mitochondrial DNA. Mitochondrial DNA play its important role on the another side. If we don't have the nuclear DNA, then we will go with the mitochondrial DNA for our analysis for DNA in DNA forensics. So basically, to know the maternity lineage, because the mitochondrial DNA always transferred from mother to next generation to generation. Okay, not from the father. No, what can be the reason behind this? That mother will transfer the DNA, mitochondrial DNA from one generation to next generation. Anyone? What can be the possible reason? Anyone? Why father is not contributing in the mitochondrial DNA? Why only mother contributing in the mitochondrial DNA? What's the reason behind this? Anyone? Anyone? Okay. So mother contribute in the mitochondrial DNA because the whole cell of the oocyte, which contain the mitochondria, which contain the nucleus, which contain the cytoplasm, that is the egg cell. Okay, egg cell is participating in the fertilization. But in the male, the only nucleus participate in the fertilization. No mitochondria, no other cytoplasm. The acrosome cap, or we can say acrosomal body, or head of the sperm, okay, play and we can say participate in the fertilization. Okay, अगर आपने fertilization का पढ़ा हो, in fertilization what happened that the tail portion of the sperm just left behind outside the egg cell after the, we can say, fertilization between the two cells, sperm and the egg. Okay, that's why the mother contribute in the mitochondrial DNA. Okay, are you getting my point? Anyone? Everyone? Any doubt? Any query? So there are different type of eras in the DNA profiling. What is DNA profiling? Simple question. If you are sitting in any viva or in any interview, okay, and in any examination, there can be simple question can be asked like, what is DNA profiling? So DNA profiling is nothing but is the study of genetic genotype. Okay, genotype of different markers. So DNA profiling ऐसे ही ग्रो नहीं हुआ. It grows stage to stage, means step by step. In 1900s, it was considered as the protein or the genetic material which are passed from one generation to next generation. But but after the advancement in the techniques, okay, after the invention of the like RFLP techniques, PCR techniques, DNA fingerprint techniques, capillary electrophoresis technique, it was considered that DNA is the genetic material which contain all the information regarding the life of the individual. Okay, so while studying the DNA profiling, while studying the history of DNA profiling, there are three type of time period are there. First is the pre-DNA years, second is the first decade, third is the second decade, and fourth one is जो जो present में भी चल रहा है, okay, the post DNA period. The pre-DNA period is from 1900 to 1985. That is the era of blood grouping and forensic protein profiling. Okay. Next decade is DNA typing, in which RFL technique was, we can say, discovered by the, किसने किया था? Who discovered the RFL technique? Who was that person? Anyone? RFLP marker technique किसने किया था बच्चे? Invent किसने किया था? Who is the father of RLP technique?
Sir Alex Jeffries.
Yes. Okay, Alex Jeffrey invented the VNTR regions in 1985 to 1995, and parallelly, the Carey Mullis also invented the PCR. Okay, that is polymerase chain reaction. Okay, so these are the questions which are always come into your examination, like चाहे वो fact का हो, if it is from UGC NET, if it any other state examination. Okay, if you are giving an exam in your DNA. Okay, these are the questions. Okay, so second phase of DNA testing is 1995 to 2005, era of DNA sequencing, where the whole technique was totally described by the scientists, and then the era of DNA sequencing was there in our life. Okay, where we are using now STR markers. Now, what are the STR markers? These are the short tandem repeats. We are not using the VNTR markers. Okay, so first of all, pre-DNA years. 1900 to 1985. In 1900, the Carl and Stinner discovered the clumping of blood and got Nobel Prize in 1930. But who discovered the blood groups? Who discovered the blood groups? Four type होते हैं ना? A,
Sir
B,
William
AB, and
Carl
O. There
Landsteiner
are different type
discovered.
of blood groups is there. Okay. Carl and Stinner discovered the different type of blood groups according to the clumping of the blood. Okay, in 1900s. But there are different type of blood groups are also there, like RH factor, MN, Duffy. Okay, ऐसे कुछ और system भी है. But the main we will discuss about the ABO blood grouping system. Four type of blood groups are discovered, like A, B, AB, and O. A is present in the 42% population out of the 100, B in 12 population, 12%, AB in 3%, and O in 43% of the population. Okay, so what was the basis, what was the principle of the, we can say, blood grouping? कैसे करते थे वो? It was based on the clumping of the antigen and antibody. It mean basically in simple words, antigen and antibody interaction is there. Okay, antigen antibody interaction is there, and the complementary antibody and antigen just clump to each other and form we can say B-like structures form of it, and it will like we can say form a agglutinates. Okay, and the reaction between antigen and antibody is called agglutination. Okay, and this is the concept of antigen polymorphism. Antigen polymorphism. What is the meaning of antigen polymorphism? Because there are different type of antigens are there in on the surface of RBCs in humans. Okay, we are discussing about the humans here, not wildlife forensic. Okay, so on the RBCs, on the red blood cells, on the surface of red blood cell, there are some specific type of antigens are there present on the surface of RBCs. Okay, so that antigens give the blood group to that individual. If a person is of A blood group, then the antibody, then the person will have the B type of antibodies. Okay, means वो carrying the opposite रहेंगे. Okay, if the person has A blood group, A antigen on the surface of the RBC, then the antibody is of B type. Okay, because while we are performing the blood grouping technique, okay, we just do क्या करते हैं हम? In that reaction, we just add a simple blood cell which contains the antigen, and उसके ऊपर हम क्या डालते हैं? On that we will add the antibodies or antisera. Okay, if it is of A blood group, then it will clump with the antibody B or antisera B. Or antiserum, not antisera. Antisera and antiserum is the two type of different type of plural and singular terms. Okay, antisera and antiserum is the singular and antisera is the plural. So next the Leon lattice. developed the method for blood grouping for the first time AVO type using in the paternity dispute in the Europe and US. There was a case of the paternity dispute. Okay, then the scientist named as Leon Lattice. So he developed a method of aviotyping from the, we can say, fresh blood. Okay, that is the direct technique. There are different type of techniques are there. Blood grouping. Anyone? कौन कौन से होते हैं टेक्निक्स डिफरेंट टाइप ऑफ ब्लड ग्रुपिंग टेक्निक्स प्लीज नेम एनीवन which we are using in the forensic lab in biology section biology division which we discover the we can say क्या बोलेंगे उसको कि हम ब्लड ग्रुप निकालते हैं फ्रॉम द ड्राइड ब्लड स्टेन ओके सो व्हाट आर द दैट टेक्निक्स एनीवन थ्री होती है ना देयर आर थ्री टाइप ऑफ टेक्निक्स आर देयर ऑफ ब्लड ग्रुपिंग यूजिंग इन अ फोरेंसिक लैब नेमली एनीवन किसी को तो पता होगा ना
Liquid blood analysis.
no anyone master student please
Tekmen Takayama.
no that is the yes. These are the blood group technique. What is the confirmatory test? Okay, confirmatory test. No, these are not blood grouping techniques, but these are the confirmatory test for the blood. Tickman and Takayama. Okay, these are not blood grouping techniques. There are another after the we can see. Yes.
ABU blood grouping and RH1.
No. Which we are using in forensic lab. Which we are using in forensic lab. Anyone specialization with the forensic biology and serology? Anyone? Please unmute. Anyone? Forensic biology तो पढ़ा होगा ना आपने? Have you read about the forensic biology? In that blood की composition तो पढ़ी होगी. What is the composition of a blood? Anyone? Then फिर ही हम आगे जाएंगे. Please answer. What is the composition of a blood? After centrifugation, which are different layers are formed in the we can say capillary containing the blood sample. कौन कौन सी लेयर बनती है कौन कौन से उसमें एलिमेंट्स होते हैं ब्लड के व्हाट आर द कंपोजिशन व्हाट इज द कंपोजिशन ऑफ
Sir, plasma type, sir. Plasma, water.
yeah yes so there are two type of elements are present in the blood one are the formed elements and second is the liquid portion so formed elements are the cells like RBCs WBCs okay platelets this is the part of formed element and the plasma and the serum okay that different type of we can say different component of the blood plasma and the serum okay so which this is your assignment that what is the difference between the plasma and the serum serum होता है ना and plasma so what is the difference between plasma and the serum Ayush please note these questions which I am giving in the presentation okay yesterday's question was on human genome project okay so your assignment is to make a small like article like we can say notes like okay human genome project के ऊपर लिखना है and now what is the difference between the what is the composition of the blood first second please note down what is the composition of the blood first second what is the difference between the plasma and the serum okay so okay come to the next question what are the different techniques of the blood grouping which we are using in the forensic science in a forensic lab how we can get the blood group from the dried blood stain anyone अभी एक ऐसा क्वेश्चन आ सकता है आंसर आ सकता है मुझे लगता है how we get the blood group from the dried blood stain because from crime scene we can't get the fresh blood we always get we can say swab okay dried blood stain ही हम लेते हैं from the crime scene so what are the different methods of getting the blood group from the dried blood stain anyone please answer anyone forensic student हो बच्चे you should know about these techniques
Absorption elution method.
good yes these are the three techniques other two are absorption elution
Absorption inhibition.
Absorption inhibition.
third one
Direct agglutination.
Direct agglutination is the Leon lattice method. Okay, okay, ठीक है. Third and one last method is there. One last method. Anyone from Jaipur FSL किसी ने अपनी internship की हो? Anybody from Jaipur, Rajasthan who did the internship from the Jaipur lab in biology division? वहाँ पे यूज करते हैं. That is mixed agglutination. Okay, there are three different type of techniques are there which from which we are getting the blood group from the dried blood stain. That is the absorption, elution, absorption, inhalation, mixed agglutination. They are very important one. Okay, so your next question for assignment is that you have to make a small brief note on the procedure of absorption, elution, absorption, inhalation, and mixed agglutination methods. Okay, got it. So Leon lattice ने develop किया. After that, and there was another system were also discovered like MN and RH factor. Okay, then there is a branch of the forensic science that is known as the forensic serology. So forensic serology is a term used for the describe the laboratory test that examine the specific antigen antibody interaction. So the study of antigen antibody interaction, we can say the study of antigen antibody interaction is called the forensic serology. Serology basically deal with the liquid part of the, we can say liquid part of the body. Okay, so that is the forensic serology. And what is the forensic biology? Simply question आपको मुझे वाइवा या इंटरव्यू पूछ सकते हैं. The simple question can be asked in your viva and interviews, which I am using both languages. Okay, English and Hindi. Okay, for convenience of every student. Okay, if you have any doubt from any point, any part of the presentation or slide, you can please ask me. Okay, by unmuting yourself. Okay, so forensic biology is a we can say branch of the forensic science which deal with the biological samples such as hair और कौन है hairs, bones, teeth. Okay, that etc. There are so many like we can say large list of the forensic biology samples. So this is the blood groups. So there are different type of blood groups are there, A, B and O. So they are different type of antigen polymorphism. If a mother has A blood group and a father have if a mother have A blood group and father have A blood group, then it will show the A blood group or O. Okay, so there are the three these antigens A, B and AB. Or simply we can say that the A and B are the dominant alleles. Okay, A and B are the dominant alleles and O is recessive. So whenever there is a recombination between the O antigen, then it will form the O blood group. What in simple, if there is a recombination between the A and O, B and O, A and B, then it will show its dominant character. The blood group will be of A, B and AB respectively. Is it clear to all? Is
Yes,
it
sir.
clear to all? Everyone? Okay. So come on the main, that is the protein profiling. So after the we can say invention of the blood grouping techniques, it was we can say discussed there that I think blood is not a part of, we can say blood can be used as a visualizing factor, but not in every case. Okay, like एक बच्चे ने क्वेश्चन ही पूछा था अभी in the beginning of the session कि in 10 to 20-year old case what we'll do. Okay, so in that case we can't have blood. Okay, अपने पास blood नहीं होएगा. Then they were trying to like make the profile based on their proteins. So proteins have a different proteins make the enzymes and every portion of the body proteins. Okay, जितने भी enzymes है, all the catalysts are there present in our body which regulate the biochemical reactions. Okay, so that all are controlled by the proteins. So next protein forensic protein profiling is based on the amino acid sequences. There are 20 type of amino acids, and that 20 type of amino acid will form the different type of sequence which individualize the individual to individual. Okay, the function of that amino acid remains same, but the sequence of that codons can be, or we can say the repeat of that codon can be vary from individual to individual. So, what is the forensic protein profiling? It is based on the amino acid sequences. There are 20 type of amino acids, and that 20 type of amino acid will form the different type of sequence which individualize the individual to individual. Okay, the function of that amino acid remains same, but the sequence of that codons can be, or we can say the repeat of that codon can be vary from individual to individual. So there was a concept, protein profiling was based on the concept of multiple protein polymorphism, because protein have different type of sequence of amino acids, so there are number of proteins are there. Okay, so there are number of type of isoenzymes. Iso means same enzymes. Okay, ये same तरह के enzyme भी हो सकते हैं body में. There are different type of enzymes are there. They will show the polymorphism. Okay, so multiple forms of the protein enzyme that can catalyze same biochemical reaction in spite of slightly difference of amino acid sequence. Okay, what are the messenger enzymes which we can say control or catalyze the same kind biochemical reaction in spite of slightly difference in the amino acid sequence. Okay, this difference in amino acid sequence will, we can say, gives to the individualization factor. So to segregate, or we can say to make the profile of proteins, we use some techniques such as starch gel, agar gel, polyacrylamide gel electrophoresis used to separate these proteins. Okay, so your next assignment is, next question is, you have to make a brief note on starch gel, agar gel, and polyacrylamide gel electrophoresis. Okay, what is the difference between these three techniques? Starch gel, agar gel, polyacrylamide gel electrophoresis. Okay, so by 1980, isoelectric polyacrylamide gel electrophoresis, and next technique, next advance with the advancement of time, there was some we can say add some part of in the polyacrylamide gel electrophoresis, that is isoelectric foxing, which used to which has a higher resolving power than the protein electrophoresis because of sharper bands. Okay, then this technique was now combined with the silver staining technique. Okay, a fluorescence provide करेगा after the getting the results, all the markers. Okay, all the marker protein profile will now fluorescence, give the fluorescence. Okay, so later on some techniques like used for AD1, SAT and early STRs. Okay, so parallelly क्या था कि some like markers like D1, SC, this is the marker. Okay, and some early STR was used. Okay, to make the profile. Okay, in the forensic science. So there are different type of isoenzymes are there. So characteristics of the sum of the isoenzymes used in the forensic protein profiling in 1980s. बच्चे आज के में इसको यूज नहीं करते. Okay, nowadays this protein profiling technique is not used. हाँ, in research field it can be used. Okay, it may be used. Then erythrocyte isoenzymes are there. There is phosphoglucomutase. There is protein symbol PGM. Number of alleles is. Just wait a minute. Okay, I have a call. Okay. Hello, am I audible? Hello.
Yes, sir.
Yes,
Yes,
sir.
sir.
Okay.
Yes, sir.
Okay. Okay. Sorry for the interruption. Actually, I had a call, important call. Okay. Hmm. So, uh, presentation visible है? Is presentation visible to everyone?
No, sir.
No, sir.
Hello, am I audible?
No.
Hello, I'm not audible.
So now it's up.
My presentation is not visible.
So it's visible and you're
Not
audible.
visible.
So now
Now
it's
it's
visible.
visible, sir.
Now it's
Okay.
visible, sir.
Okay. Okay. Good. Good. Okay. So we are starting from it. So there are different type of, we can say isoenzymes of the protein in protein profile is used for. So okay. So there are different type of, we can say isoenzyme. These isoenzyme are nothing but act like a markers. Okay, marker की तरह use करते थे, and there are different type of number of allele and there are different number of, we can say, discrimination potential, like 10 phenotype possible with IF, then 0.67%. ऐसे करके we have a different like characters discrimination potential for different type of protein. Okay, in protein profiling. So next, so that was about regarding the uh, pre-DNA year. Okay, from 1900 to 1985. Then when the Alex Jeffrey invented the VNTS and a technique that is RFLP, okay, was, we can say, discovered. So it, we can say, एक revolution आ गया बच्चे. in a forensic field, in a forensic DNA analysis. Okay, so with that, we can say RFLP technique. Now DNA is used as a genetic marker or genetic marker. We can say genetic material. Okay, so this is the first decade of the DNA typing, that is from 1985 to 1995. Okay, so Alex Jeffrey 1985 worked with the VNTR. That is VNTR are the variable number of tandem repeats using RFLP DNA testing technique. RFLP means restriction fragment length polymorphism. Restriction fragment length polymorphism. Now every single word has its significance. What is restriction? Why it is so called restriction fragment length polymorphism? Because in this technique we are using some enzymes, some restriction enzymes, which cut the DNA from specific fragments along the length of the DNA, which show the polymorphic nature. Okay, so these four words are explained like that: RFLP, restriction fragment length polymorphism. That is technique. Restriction enzymes are used to cut the fragments along the length of the DNA, which show its polymorphic nature. Then Weaver and White, were the two scientists, show the RFLP marker are the polymorphic in nature. Okay, polymorphic means they can individualize from person to person, individual to individual. Then these markers follow the law of Mendelian inheritance across the generations. Okay. Next, the results of the multi-locus probe look similar to the barcode. Okay, barcode देखा होगा. Have you seen the barcode? So that the resulting profile is look like a barcode. Okay, then this pattern is told as DNA fingerprints, because DNA fingerprints are unique to each other or unique to in every person. Okay, unique in every person. So this, we can say, resulting DNA profile is unique from person to person. So that's why known as, it's known as DNA fingerprints. Okay, so DNA fingerprints are so called because the variable markers are unique and give a unique pattern of each individual. Jeffrey teamed up with the Petrogel and David. ओके, इनके यहाँ इन्होंने टीम अप किया. डेविड रेट फर्स्ट टाइम I publish a complete journal on the forensic application of DNA fingerprint in the December 1985. Okay, first journal इन्होंने publish किया in 1985 on the DNA fingerprints. At the same time, Carey Mullis invented a new technique, that is the PCR. PCR is the polymerase chain reaction to make the copy of any desired region of the DNA. Now PCR is very important in DNA amplification or DNA profiling. Without PCR, we can't get the profile of desired DNA fragment, because PCR make the 100 to 1000 copies of desired fragment of the DNA. Okay, so who invented? Carey Mullis. Okay, so अभी हम पढ़ेंगे ना, we will discuss what the RFLP technique in detail. RFLP based DNA typing. So this technique was given by Alex Jeffrey in 1985. Technique RFLP, genomic DNA preparation, which there are different type of, we can say, steps, okay, involved in the RFLP technique. First step is the genomic DNA preparation. Genomic DNA preparation means extraction of the DNA from different samples such as blood, hair, okay, bone, skin, other tissue samples, saliva, semen, sweat. Okay, then restriction endonuclease digestion of the genomic DNA into the fragment. But these restriction endonuclease, there are two type of enzymes are there. Okay, one are the endonuclease, second कौन से होते हैं? Second are restriction endonuclease digestion enzymes. There are two type of restriction enzymes are there. One is the endonuclease. Second, anyone? Okay, this is your homework question. Okay. तो आपको डिस्ट्रिक्शन एंजाइम के बारे में पता होना चाहिए. ओके, यू नीड टू like make a small chart, like, okay, that regarding the restriction enzymes. Okay, so restriction endonuclease enzymes are the enzymes which cut the DNA from a specific fragment. Okay, so these restriction enzymes again have its we can say a seizure. These restriction enzymes behave like a seizure, biological seizure. Okay, because these restrictions then contain the specific type of, we can say, sequences like GC, 80, ऐसे करके. So they contain a different type of, we can say, sequences which we can say have the after the we can say denature the DNA, they just combine with the DNA molecule of the host, and it will make the we can say cut the DNA from that fragment. Okay, and form the flanking ends or sticky ends for the primers. Okay, primers are the we can say small synthetic or we can say artificial human made markers which are just we can add into the our reaction mixture. Okay, next hydrogel electrophoresis. After the cutting the DNA into different type of fragments, we use hydrogel electrophoresis for the separation of DNA fragments according to their size. Electrophoresis तो आपको सभी को पता होगा. I think you all know about the this technique gel electrophoresis. Okay, agrogel electrophoresis. Then after the separation of DNA fragment according to the size under the influence of electricity in electrophoresis, the DNA fragment then transferred to the nylon sheet. Okay, using southern blotting technique or southern transfer. Southern was a scientist name, and he gave this technique, so that's why known as southern blotting. Then after the transfer, we need to add some primers or we need to some artificial which we need. Okay, or we can say desired primers then added to the reaction mixture. Okay, where the hybridization with the local specific form takes place. Okay, then last is the detection. Okay, last step is the detection of the local specific band by the autoradiography and or chemiluminescence. Okay, so these techniques took several weeks to complete. Okay, here is some correction. Okay, this technique takes several weeks to complete. So process क्या है बच्चे? Just simple, very simple process. पढ़ने में only just to read. The process is very simple. But while we are performing this technique, very tough. Okay, because DNA always weak contamination free. Weak contamination नहीं होना चाहिए बच्चे. If there is a small contamination is there in the sample, then it will lead to the mistake in the DNA profile. So process simple. First of all, you are going to the crime scene. You are collecting the sample, desired sample such as blood sample, tissue sample, hair sample, any other sample. Okay, we just select at the crime scene. Then after the collection of that sample, you need to break. Then it will submitted to the law enforcement agency, and law enforcement agency then submitted to the forensic lab. After submitting to the forensic lab, like or we can say the division, give a specific number of that case. Okay, this is the case of 120. This is the case number 130. This is the case of 140. Okay, accordingly numbering होती है, and after that opening of the case is there in the forensic lab. After opening, first of all, a scientist or we can say a forensic expert need to read the cautionary given by the IO. IO means investigating officer. Okay, then after that they are performing their own lab work. Okay, lab work में क्या क्या रहेगा? In lab work includes the extraction of DNA. So first of all, breaking down cell membrane to extract the DNA from the nucleus. Okay, so second step is the cell lysis to break the cell membrane and moving the packaging of the protein around the DNA. That is DNA extraction. Then addition of restriction enzymes. That is the biological seizures that are capable of finding and cutting the specific DNA sequences, so that extracted the DNA molecule fragments into smaller pieces. Okay, then here note that is the most commonly, but there are two type of restriction, and there are different other type of restriction are also there, which are वो जो हम use करते हैं forensic, which we are using the forensic DNA analysis. We use the thermolabile. Thermolabile means which can withstand in the high temperature condition. So the most commonly used restriction is HAE3, which requires GC rich site, GC rich, and cut the DNA fragment and stick here. Okay, HAE3 again, but इनका nomenclature different होता है. To give the we can say name to the different type of restriction enzyme. Clear? So there is a different type of nomenclature. Like HAE means hemophilus. Egyptus is the name of bacteria. 3 is the sequence. The number of that number of sequence can enzyme is extracted from that bacteria. Then the chopped DNA fragments then separated according to their fragment size in the agrocell electrophoresis. Then next is southern blotting. Southern blotting means named after the Sir Edwin Southern 1970. Separated DNA fragments then transferred from the gel to the solid matrix, nylon membrane. After under the alkaline condition by treating with NaOH, as a result, DNA become single stranded DNA and denatured DNA. Okay, so in southern blotting technique, after the separation of the DNA fragments, when we transfer the fragmented DNA in onto the nylon membrane, so with the help of the UV light, it will cross-link to the nylon membrane. And here the under the alkaline condition, the DNA is in the denatured condition. Denatured means single stranded condition. Okay, so the DNA then immobilized by nylon membrane by using UV ultraviolet which radiation डालते हैं, it will cross link to the membrane. Then the DNA strand fixed to the membrane by cross linking the DNA onto the membrane with UV light. After that, hybridization of DNA with radioactive probe. Hybridization means hybridization word we have heard in the chemistry or in chemistry है ना, hybridization of the orbitals क्या होते हैं, वो just chemistry का part है, you just read in the chemistry, but hybridization term here used to describe a new product will form, or we can say from the parent DNA fragment a new primer will add up to the parent DNA fragment, and where the a double stranded DNA is formed, but that DNA fragment is of our choice. Okay, that is the desired DNA fragment. Okay, so the VNTR radioactive probe, we can say, added. These radioactive probe are the artificial, human-made. Okay, which in which us we can say a radioactive substance or radioactive element hota hai, is added to the we can say a small fragment, and it is put up into the reaction mixture, allowed to hybridize to the single stranded DNA under the optimal temperature and ionic strength, and labeled the probe to find out their complementary sequences, and which it will run through the along we can say along the length of the DNA fragments, and where there is a complementary sequence, it will just bind to that VNTR region of the parent DNA. After that, washing of excess of probe from the nylon membrane. Okay, then washing करेंगे. After the washing, we will detect the that X-ray film. Okay, under the we can say X-rays. Okay, we will detect that fragmented, or we can say barcode-like structure is can be detected or visualized under the X-ray film. Okay, formation of photoradiogram then reduced to the scanning and comparing with the band position. Then we have to compare. Okay, so this is the simple diagrammatic view of the overall RFLP technique. First of all, restriction digestion, we have to cut the DNA into small fragments. Then we have different type of enzymes such as HAE3, okay, which will cut the GCGC region. And then another type of fragments are also restriction enzyme are also there, such as HIMP1, PST1. Okay, HIMP1 will cut the DNA from this sequence GA. Okay, and DC. Okay, this we can say are the sequences. Okay, then restriction, these are restriction sites. Then DNA on the exogel. Okay, we have to denature under the alkaline condition. Then we have to transfer to the nylon membrane while using a southern blotting method. Okay, then we have to like cross link the DNA fragments on the nylon sheet by using UV cross linking technique. Okay, then after that, detection of the axial film. Clear? Is it clear to all? Any doubt? Any doubt in this technique? RFLP? Anyone? Any doubt?
No, sir.
Okay. And if you want to read about the this technique, just take your 12th class book. Okay, and that there is a unit is there biotechnology. I think biotechnology की unit है, and there बहुत ही अच्छे से RFLP को explain किया गया है. Okay, in 12th class book biology. So there are different that time two techniques were invented. One is single locus probe technique, next is the multi locus probe technique. Single locus, in single locus we just take the only single marker. but in multilocus we can make the different type of, we can say, we can different analyze the different markers simultaneously. Okay, six of the SLP single locus probe RFLP markers used during the 1980. Okay, six SLPs, that is single locus probe, was used. The probe only hybridized to the single VNT locus, and technique is called a single locus probe. The SLP generates a simple profile called DNA profile consisting one band for the homozygous and two band for the heterozygous. Okay, समझे, understood this? कि one band for homozygous. Okay, in nowadays if it is a homozygous condition, बच्चे अगर आपने कभी DNA profile देखा हो, in that DNA profile you will see the peak. Okay, peak बनेगी. Okay, if it is a homozygous condition, it will give it the large peak, and if it is a heterozygous condition, it will form the two small peaks. Same that. Okay, same उस चीज का बच्चे principle लिया है in that one band for homozygous and two band for the heterozygous condition as per the locus. Clear? So that is multilocus probe technique. The multilocus probe technique can detect multiple VNTR loci simultaneously. Some VNTR are GC rich sequence of 10 to 15. These four sequences hybrid to multiple VNTR and as a result of code-like complex pattern will formed. Okay, a barcode-like structure is formed there in multilocus probe. Okay, we can use multiple VNTR marker here. So this is, there are different advantages also there. So MLP technique is called as DNA fingerprinting. This technique is called as DNA fingerprinting technique. So there are different types of advantages are there, such as high cover discrimination, method was used for the patent testing in immigration dispute with greater success. But there are disadvantage also. The interpretation of mixed DNA sample from more than one individual is nearly impossible due to complex DNA fingerprint pattern. The mixed, in simple words, we can say that the mixed DNA sample can't be analyzed. Okay, is it clear to all? What single locus and multi-locus probe? So this is this simple diagrammatic view to the single locus and multi-locus probe. With single locus, we have probe 1, 2, and 3, and multi-locus we use multiple VNTR and a one, we can say, one slide. Okay, you कुछ भी आप बोल सकते हो, we can say plate. Okay, so what are the restriction enzymes? यहीं पे आ गया आपका topic. Okay, so what are the restriction enzymes? These are the proteins discovered in various bacteria that function as smart biological scissors cutting the DNA molecule precisely as a specific sequence pattern. Restrictions enzymes are play a very important role in the, we can say, DNA, not in the forensic things, but also in the research field, like in biotechnology, biological methods, in plant and botanical researches, okay, or in DNA forensics. These patterns are palindromic sequences. Palindromic, what are the palindromic sequences? Anyone? What are the palindromic sequences? Anyone? किसी को पता होगा? Anyone? Palindromic sequences. Am I audible to everyone?
Yes,
Yes,
sir.
sir.
Hmm. Just interactive session. If you are responding to my questions, just yes or no, it will make like a more interactive session. Okay, interactive होता है, which will like give me motivation. Okay, so you need to respond with every question I ask you. So what are the palindromic sequences? Anyone? Yes or no?
Sir,
You know? Hmm.
yes, sir. Sir, palindromic sequences is the DNA sequences in which nucleotide sequences is the same when it's read in like 5 strand to 3 strand and direction on both
Hmm.
complementary strands, sir.
Good. 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 Already written over there on the slide. Okay, you can just take an example of take an help of this slide. So palindromic sequences are the sequences which can read same in both the direction 5 to 3 and or 3 to 5 and. I'm just taking an example, that is nitin. The name is nitin, Nitin. Okay, you can like make the analogy for that. Nitin, Nitin. Nitin can be read from left side or from the right side. Both sounds the same. Sounds the same. Okay. So restriction enzyme cut the DNA strand to generate sticky ends which have a overhead of few nucleotides. Restriction enzyme कैसे cut करेंगे? It will cut the DNA strand over the few nucleotides sequences. to make the sticky ends, to generate the sticky ends. The restriction enzymes cleave the phosphodiester bond between the DNA at a specific site known as restriction sites. So it will only act on the 4 to 8 base pair. Okay, that are the restriction sites. Okay, because the restriction enzyme is means on a 4 to 8 base pair a nucleotide we can say. Okay, restriction में खुद भी 4 to 8 base pair एक बच्चे होता है nucleotide होता है. Okay, it will just act on a specific site of the DNA and cut that site. Okay, for the sticky ends to generate the sticky ends. So these three type of restriction enzymes are just wait. I have to remove my lective line. Just wait. Just wait a minute.
Just wait, everyone. So we'll be back in few minutes.
Am I audible?
Yes, sir.
Am I audible? Hello.
Yes, sir.
Yes, sir.
Hello. Okay. Okay. So there are three type of restriction enzymes are there based on their subunit composition and geometric properties such as type 1, type 2 and type 3. Okay. So another type of enzyme important. Okay, type 2 enzymes are more important, commonly used and require magnesium as a cofactor, Mg2 positive ions. Okay, so like Ha3 is from Haemophilus aegyptus and 3 stand for the third endonuclease isolated from the bacteria. Okay, clear. Then detection. How we are going to detect the DNA probes? What is the detection technique? Nowadays same techniques are used, but in an advanced way. But in an advanced manner. Okay. So to detect the VNTRs, labeled SLP probe is hybridized to a target sequence DNA and become immobilized to the nylon membrane. So any unbound probe we have to wash away. Okay, we need to wash the unwound probes. Okay, because it will if we don't not wash the unwanted probes after the we can say this hybridization. Okay, it can we can say interfere with the results. Okay, so radioisotope labeled with the phosphorus 32. Okay, can be used. The hybridized probe can be detected by exposing the membrane to the sheet of the X-ray film to generate the autobiograph. Oh, sorry, autoradiographs. Okay. Alternatively, there are different detecting techniques also there. Was also there that enzyme conjugated probe can be used. Okay, in which a specific type of enzyme such as alkaline phosphatase is an enzyme that can be detected with the chemiluminescent substances. So a chemiluminescent substance can be detected by exposing to the X-ray film. Okay, chemiluminescent substance कौन सा होएगा? The chemiluminescent substance is that substance which will give the luminescence. Okay, like fluorescence or phosphorescence. Luminescence is further of two type: fluorescence and phosphorescence. Okay. Next question is you need to make a brief note on luminescence. Okay. Note करते हैं ना. Next, long range PCR. In this decade, now PCR is also going, we can say, used parallelly with RFLP technique. In late 1990s, an attempt was made to develop a ability to use PCR amplification of entire VNTR region. Okay, जितना भी VNTR region है, the whole VNTR region was we can say made attempt was made to we can say copy or amplify the entire VNTR region, but no good results were found. Okay, because VNTRs are very long sequence sized markers. The technique involved the VNTR region with the mixture of tag polymerase. Now, tag polymerase is another we can say enzyme, a thermostable enzyme which we use in the PCR. Okay. So according to the examination point of view, that a lot of time a question is asking you a UGC NET fact and fact plus examination, then which enzyme we are using in the PCR, that is tag polymerase. Now, what is the full form? What is why it is so called tag polymerase? And what are the different characteristics of the tag polymerase? These are the thermostable enzymes. Can we can say will stand in the high temperature. So the different type of we can say markers were discovered. Okay, along with the PCR and with the help of PCR, there was attempt to amplify these sequences like HLA DQ alpha or DQ alpha. HLA means human leukocyte antigen. ओके लिक्विसाइड एक क्या होता है पार्ट होता है ना ब्लड का सो लिक्विसाइड एंटीजन इट इज अ मार्कर प्रेजेंट इन द ब्लड सो ह्यूमन लिक्विसाइड एंटीजन दैट इज DQ alpha. The first PCR based DNA test kit was detecting sequence based on the human leukocyte antigen that is HLA DQ alpha gene found on the chromosome number 6. chromosome number 6 के ऊपर ये वाला मार्कर होता है and this was given by that kit was given by the Citrus Corporation, a company. Okay, list purchased by Perkin Elmer. Okay, which if you will like work in the forensic lab, DNA lab, you will mostly go through the some names like उसका किट चाहिए हमें उसका किट चाहिए है ना we need a Citrus Corporation kit, we need a Perkin kit. Okay, we need Quaison kit. Okay, nowadays Quaison kit is mostly used. Okay, in forensic lab. So that kit contains all of the component which we need in the DNA amplification of DNA analysis of DNA profile. Okay, so that kits cost for I think 1 crore to 80 lakh, 10 lakh, 20 lakh according to their we can say specificity. So released the DQ alpha ampli type kit in 1990s. The first DQ alpha kit could be distinguished 6 markers. जो first DQ alpha kit थी which is that distinguished only 6 markers. That is 1.1, 1.2, 1.3, 2, 3, 4. Okay, only on 6 chromosomes. Okay, these are the chromosomes. Okay, 1.1, 1, 2. With these 6 alleles, it was possible to define 21 genotype. Clear? Then after that, polymarker kit was developed. Okay, that is PM plus DQ alpha along with DQ alpha polymarker. Okay, with the same kit, we add some more markers. Such a recognize the need for greater power of discrimination. This kit was given by Perkin Elmer. Okay, developed as a new ampli type EMDQ alpha kit. Nowadays we are using STR kits. Okay, STR kits होती हैं. This kit also referred as polymarker co-amplified with the HLA-DQ alpha. Okay, this polymarker kit is co-amplified with the DQ alpha. We उनको combine कर दिया. Okay, just combined both the kits. Okay, and give the more power of discrimination along with some more chromosomes such as 4, 7, 11, 19. It was only capable of power of discrimination in the range of 1 in 10,000 unrelated individuals. Okay, so these are the some characteristics of DQ alpha and polymarker loci used in the ampli type PM kit in the 1990s. So DQ alpha gene product, HLA DQ alpha chromosome number location अभी आपको पता होगा. Now you know D6P 21.3 क्या है. Okay, number of leads are there or 7 leads are there, heterogeneity 0.8. Okay, so this chart we can say given by that company. Okay, the different type of markers are there. Now after that D1S80 PCR amplified VNTR invented. Students listen, ये sequence है ना, the sequence of we can say development of different DNA kits or different markers of DNA. Okay, you have to remember this sequence in a sequence. Okay, means ऐसा नहीं कि पहले आप D1 SAT याद कर रहे हो after that second. No, you have to remember in a sequence because in examination as per your examination point of view, they ask the sequence of the development of the different type of markers. Okay, like a question like that, arrange the following DNA markers DNA kit according to the sequence in ascending order, descending order. Clear? So D1S80 PCR amplified VNTR. The amplified fragment length polymorphism, or we can say simply in a simple short form, AMP FLP. Okay, that gained the greatest use in the early 1990s was D1S80. Okay, a mini satellite. Now, a term is used as mini satellite. On chromosome number 1, used containing a 16 base pair and 14 to 41 repeats unit. Okay, this D1S80 contain 16 base pairs, and how many repeats are there? 14 to 41. Okay, now every individual have different, we can say, repeat. If a person have 14 repeats, a second person can have 15 repeats, a third person can have 16 repeats, fourth person can have 17 repeats. Okay, so this will distinguish, or we can say this will individualize person to person. D1 SAT range from approximately 400 to 800 base pair and were typically separated out vertically polyacrylamide gel electrophoresis technique by silver staining detection technique. Okay, polyacrylamide gel electrophoresis आप करके ना, you have a homework that you need to differentiate between these three techniques: starch gel, acros gel, polyacrylamide gel electrophoresis, okay, and silver staining techniques. Then in some cases, D1 acid were combined with sex typing markers such as X and Y. Okay, these as D1 acid marker then co-amplified with the amylogenin. Amylogenin is a sex marker. Okay, which will like have the X and Y alleles. Okay, the size of the X and Y alleles, X has a 2 1 2 base pair and Y has 2 8 1 base pair respectively. Okay, clear? Clear, बच्चे?
Yes,
Any
sir.
doubt?
No, sir.
Okay, good,
No.
good.
No, sir.
Okay, good. So now we are going to next advancement in the DNA forensic, that is STR. The VNTR था. If we have VNTR, then why we need STRs? है ना? Why we need STRs? Because these STRs so called as the minisatellites have a repeat of 41 base, sorry, 14 to 41 repeats are there. Okay, so STRs would be used as a genetic marker in the futures. Now we are using STR markers. Okay, in some labs, 28 markers STR marker are being used, or in some labs, 22 markers are used. In some labs, 20 markers are used. Means different lab have different kits, so different kits contain different type of markers. Okay. So STR markers have a number of advantages. First of all, STR markers can be amplified by PCR because of their small size. Now just remember that time. Okay, VNTR were also to amplify the VNTR. The attempt was made with the help of PCR. है ना? But that attempt was failed due to the long size, or we can say large length of the VNTR. But this limitation was overcome by the STRs. The STRs can be amplified with the PCR because of their small size, high rate of sensitivity. STRs are highly variable. Okay. In 1995, USA has selected 13 STR, and that 13 STR is known as CODIS. Okay, combined DNA index system. Firstly, we can say given by the USC lab. Okay, so 13 STR CODIS system बनाई गई सबसे पहले, and that coding system combined DNA index system contain only 13 STR. Now nowadays we are using double to that 13 STRs. Okay, so first commercial STR kit available from the Promega Corporation. Okay, there is another company name, that is the Promega Corporation 1994 for silver staining analysis. Okay, this kit consists of STR marker like CSF1PO, TPOX, TH01, named as CTT. This kit was named as CTT. CTT means triplex using the first letter of each locus. Okay, okay. C from CSF1P0, PO, sorry, T from T-box, and second T from TH0. Okay, the kit was named as CTT. Okay, now you can see the difference here. Mini satellite, micro satellite. Mini satellite, you can see VNTR, or micro satellite you can see STR. So mini satellite is 16 base pair, and what STR is 4 base pair repeat unit. Okay. Now we just end here the first decade of the. We ended here pre-decade, first decade. Now we are on the second decade of DNA testing. Now the second decade, which is after the invention of STR, after the invention of PCR, after the invention of like proper staining techniques. Now in second decade, it was a big challenge to detect the DNA profile. Okay, so different techniques was, we can say different techniques were discovered or invented. Okay, to just detect the DNA sequences of DNA profile. So सबसे पहले क्या है कि first of all we are discussing about the silver staining STR kits. Silver staining procedure was used for the commercially available STR kits from the Promega Corporation. Okay, same again, again and again. You will read about the company's name: Promega, Citrus, Quasar, etc. and all. Okay, so these companies provide you a kit. Okay, proper kit provide करेगी, and that kit contain the reaction mixture for extraction, reaction mixture for PCR, reaction mixture for genetic analyzer. You just need to add the sample in that reaction mixture, and you have to put that vials in the, we can say, machine, and after that the machine will perform all the work. Okay, no laborious work is at all, and nowadays in DNA. Okay, all work is done by the machine. Okay, machinery based work हो गया in DNA forensics. Okay. So silver staining procedure are used for the first commercially available STR kits from the Promega Corporation. The silver stain detection methods are still quite effective for the laboratory that want to perform DNA typing from a much smaller startup cost. Okay. Why it is so used? Because it is need a small startup cost. No expensive instrument are needed. Simple gel box or electrophoresis or silver staining, silver nitrate. Okay, silver staining means silver nitrate is used for the developing, or we can say for detecting the DNA profile. So in this technique of silver staining, gel transferred from various pans filled with the staining solution of the stain to stain the DNA. Okay. In this technique, we need to transfer the fragmented DNA, or we can say hybridized DNA, from the various containers or various pans. Okay, to stain the DNA. First, gel is immersed in the solution. Okay, that gel contains DNA fragment. Okay, then gel is immersed in the solution of silver nitrate, that is 0.2%. Then a silver bind to the DNA and then reduced to the formaldehyde to form a metallic layer on the silver over the DNA. Okay, in gel. Then photograph taken in silver stained DNA stranded in the gel for the permanent record, and then silver staining is a lesser hazardous. Okay, because it is not cancerous. किस advantage है? Okay, but in some labs, ethidium bromide एक dye होती है, ethidium bromide dye was used for the, we can say, visualization of the DNA. But ethidium bromide had demerit. Ethidium bromide is cancerous. Okay, it can cause a cancer just by touching your finger. Okay, it can absorb through the skin and cause a cancer. Okay, so that's why silver nitrate was used. Okay, that is another radioactive reduction, that is actinium bromide. So next kits were fluorescent detection kits. Okay, so in fluorescent detection kits, we just add a element which give fluorescence like phosphorus. Okay, different type of isotopes are there. Okay, which give phosphorus. Now luminescence is just nothing. It is just excitation of electron from the inner orbit to the outer orbit. And when that excited electron just, we can say, coming from the outer orbit to the inner orbit, okay, at different levels, it will emit the lights. And that lights, we can see fluorescence or phosphorescence. Okay, so one of the first STR multiplex to be developed was the quadruplex. Okay, which again a kit was formed, created by Forensic Science Services, that comprise. Okay, again company name है Forensic Science Services, that comprise of 4 loci, that TH01, FPS, VWA, F13A1. This is so-called first generation multiplex. Okay, the second generation multiplex made of 6 polymorphic markers, STR marker or gender identification marker. Okay, gender identification marker is again a male child. Okay, 6 were TH01, VWA, FGA, D8S1179, D18S51, and these are the markers. Okay, today multiple species like above 15 or more STR are used for routine in the forensic laboratory. Okay, I already told you that nowadays we are using 24 to 28 markers, STR markers in forensic labs. So these are some, we can say, company name: Omega Forensic Science Services or Omega Applied Biosystem. Okay, giving the different kits. Okay, kit name like CTT, Gamma STR, Amplify, STR Blue, Amplify STR Green. So it contains the different locality H01, WVWA, okay, TH01, and again, okay, Melatonin. Okay. So you don't need to learn these kits. You just name the company name Yarko, Omega Corporation, Applied Biosystem. Okay. Nowadays, Applied Biosystem mostly used like for extraction. Okay, extraction अगर आप देखोगे mostly most of the laboratories or DNA laboratories, there the company name was Applied Biosystem written over there on the genetic analyzer machine, on the extraction machine, on the PCR machine. Okay, different like company name are written over there. Just go to the forensic lab. If you are doing internship, just go to the DNA division, just note down the company name. Okay, this is your next assignment. Okay, whenever you are going to do internship in any lab, okay, long term आपकी assignment है, you need to just look over the company names. Okay, the forensic lab. So next we are discussing about the mitochondrial DNA sequencing. Clear? So now here, historical background of DNA profiling we can say that ended. Okay, but yes, after that 2005, different advancements were there. Like in mitochondrial DNA sequencing, by STR typing was there. Okay, by STR means sex chromosome typing was there, and from the tooth pulp. Okay, tooth pulp में मेलाजन होता है. Okay, and then with different type of techniques were used and different type of kits were then made within the time with the advancement of time. Clear? So now we are discussing mitochondrial DNA. Mitochondrial DNA developed to enable the recovery of DNA results from the bones, hair, and teeth. So basically, mitochondrial DNA can be present in the cell or used for the lineage linkage. Okay, maternal lineage linkage, because that mitochondrial DNA can be transferred from mother to her offspring. Okay, mitochondrial DNA can be transferred from any cell. Okay, you can extract from here, root hair, okay, means rooted hair, or any tissue, any bone, any blood. Okay, means in which the mitochondria is present. Mitochondria is cell organelle, which is powerhouse of the cell. So, use because of multiple copies. Why it gives so much importance? Because a cell contains hundreds of multiple copies of the mitochondria, and mitochondria contain 100 copies of the mitochondrial DNA. So mitochondrial DNA analysis can be involved in DNA sequencing. The primary extension method was sequencing known as Sanger sequencing. Okay, but after the 2005, the sequencing methods were then going to be like introduced in the DNA and the era of DNA. Okay, like Sanger sequencing, and for that, uh, Fred Sanger like got the Nobel Prize. Okay, who gave the Sanger sequencing, who was first described in 1977. Okay, now we are going to discuss the Sanger sequencing. What is it? Is simple. Okay, not like going to the depth of the topic. This is Sanger sequencing. Nowadays, we are not, it's like we are using the sequencing method which is based on the principle of Sanger. Okay, but nowadays advancement of techniques is there. So, one more thing, if you want to see the like in detailed video on mitochondrial DNA, just go to my YouTube channel, that is the Stephen Forensic Academy. I will send you the link. Okay, because in this presentation I didn't cover the mitochondrial DNA thoroughly. Okay, so you can like watch the mitochondrial DNA from the video. Okay, from YouTube. Clear? So the Sanger sequencing for mitochondrial DNA, the process involves the polymerase incorporation of the D-dideoxyribonucleotide triphosphate, that is DD-NTPs. Okay, क्या करते थे in Sanger sequencing? They just add the different DD-NTPs, di-deoxyribonucleotides, as a chain terminator, followed by the separation step capture of single nucleotide deletion. Now, In these DDNTPs, there is no hydroxyl group at the third end. Okay, hydroxyl group is, if hydroxyl is not there, then it will terminate the reaction. Okay, so there is no hydroxyl group at the three end of the DNA nucleotide with the DT and TP, and therefore chain growth terminated when the polymerase incorporate the DT and TP into the synthesized strand. So which is basically in simple language, when there is a growth in the reaction mixture of a desired DNA fragment, so we just add the DD and TP. So DD and TP, that is dideoxyribonucleic acid, triphosphate. Now it can be adenine, it can be guanine, it can be cytosine, it can be thymine. Okay. So whenever we are adding these were added just to terminate the reaction. means वो चलती ना रहे. Okay, just to terminate the reaction. Extendable DNTPs and DDNTPs terminate both present in the reaction mixture, so that some portion of DNA molecule were extended. Clear? Is it clear? So at the end of the sequence reaction, a series of molecules are present in that differ from one waste from the another. Okay. So end में क्या होएगा? At the end of the sequence reaction, a series of molecules present that differ from one waste to another. Okay, means they got separated. Four different colored fluorescent dyes were attached to the four different type of D and TPs. Okay, there are four different type of nitrogen bases are there. So we need to add four different type of color fluorescent dyes, such as DDTP thiamine is labeled with the red dye. DDCTP cytosine is labeled with the blue dye, and DATP means adenine is labeled with the green dye, and guanine is labeled with the yellow dye. Okay, so accordingly they give the signal in the reaction mixture after the, we can say, processing of a whole DNA profile. While we are detecting the DNA profile, they just give the signal, and different colors will represent the different type of nucleotides. Okay, is it clear to everyone? Any doubt up to here? Any doubt? Which is, you need
No,
to
sir.
read from a book.
No, sir.
Okay, okay. Are you getting my point? अच्छा लग रहा है session?
Yes,
Yes,
sir.
sir. Yes, sir.
Okay, good. बोर तो क्यों हो रहे? Hmm? Are you boring? बोर हो रहे हो?
No, sir.
Okay. कुछ इंटरेस्टिंग बात करी?
Yes, sir.
It is almost about 1 and 30 minutes. 1 hour and 30 minutes. Okay, 1.5 घंटा हो गए for the session. So I am giving you an case study regarding a DNA. Okay. Now we are going to discuss one case study here. Just wait. If you are a reporting officer, okay, if you are a reporting officer and you just report a DNA, means you are working in DNA division and you report a case. Okay, you reported a case. And after some time, when that case was in the trial in the court, you got the summon. You were summoned by the court. Okay, regarding that case. What when you were like doing your work, your report was like that. You just क्या बोलेंगे उसको? You have two different type of DNA profile from a single person. Okay, means you have a sample of blood sample. You have a blood sample, and you just extract the DNA from the blood, and you just make the full DNA profile. Okay, and secondly, you also have a hair sample of that person. Not hair means saliva. You can say anything apart from the blood. You have a different tissue, a different sample. I just extracted the DNA sample, just made your DNA profile, and you were surprised. Why? Because you saw that from a single person there are two different types of DNAs profiles are there. How is it possible? Are you getting my point? Are you getting my point?
Yes,
Hello.
sir.
So you were surprised that a single person, means how a single person can give a two DNA profiles. Okay, then our attempt was again made, means you thought that I think there was some load, some kind of a contamination will be there. Just repeat your process again. Just you made your profile again, and again the same results were observed. Okay, from blood and the other cell of the same person. You just have two different DNA profiles. So what can be the possible reasons regarding that case? क्या हो सकते हैं? Anyone?
Contamination, sir.
No, 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 no. I already explained here that you repeat your test. Okay, you still you have a like doubt. And there was some lot of contamination was be there. So you repeat your test, and again you score the same results. Two DNA profile was of a person. Any other? What can be the possible results? So this is your assignment work. Ayush, please take care of that. This is the case study I already mentioned in this presentation. Okay, and you need
Okay,
to
sir.
give a, okay, tomorrow you have, first of all we will discuss about this. Okay, what are the possible causes? Okay, you can just we can say Google, but if you are giving your explanation, you need to mention your reference. Okay, reference भी देना पड़ेगा. Don't go to chat GPT and ask the questions. Okay, you need to give the authentic reference also and submit it to the Ayush. Okay, if the references are authentic, then Ayush, please give that student full marks. Otherwise, cut the marks. Okay.
Okay, sir.
So this is the case study also, and this is your assignment also. You need to take a different type of possibilities which can be in there in that scenario.
Sir,
Okay. Yes.
sir, it can be possible that before they collecting the any sample of the DNA,
Hmm.
the crime person can intentionally mix some of the some other DNAs to his blood, like injecting.
Okay. Then you will get the two DNA profiles, means mixed profiles. We
Yes,
don't
sir.
get the no. Actually, I am explaining here that you got the single DNA profile.
Yes, sir.
Single DNA profile means from blood there is different DNA profile. Then from other samples like saliva, hair, or we can say other tissue, there are different DNA profile. But of a
Yes.
same person. Okay, not a mix, means not a mixed profile. If you
Okay.
like in your question, or we can say in your query, in your scenario,
Okay, sir.
that mixed DNA profile will be there. Okay, if a accused will like intentionally tamper the evidence or mix the
Okay, sir.
sample. Okay, that will give the mixed profile. Anyone else? Anyone else? You are the forensic scientists, and you have a very broad thinking because some of you will go to crime scene investigation after your completion of your studies. Some of you will do work in the crime labs, sorry, for FSL labs. Okay, scientific officer. Okay, so you have a broad thinking regarding this.
Sir,
Anyone else?
sir, one doubt, sir.
Yes.
Sir, what is touch DNA?
Touch DNA is nothing, just whenever you are touching any object and that there is a like a mutual exchange between the surface and your hand. Okay, just
Okay,
leave
sir.
your some saliva. Just while you are touching to any object, just exchange your DNA and some object from that material. Okay, that is the touch DNA. So here the Locard's exchange principle work here.
Okay, sir.
Whenever you are on any crime scene or when you are on
Okay,
any
sir.
surface, you just there is a mutual exchange between the two objects. Okay, so touch DNA is nothing. It is just whenever you are touching any object, it that's why it's so called touch DNA. Okay,
Okay,
because
sir.
whenever you are touching any object, the cells or epithelial cells, upper surface, upper layer of the skin is epithelial, na? So epithelial cells removed, we can say transferred to that surface, and then we need to collect that samples from that surface. Okay, there are a lot of researches are done on touch DNA are there, like gloves से निकालना to extract the DNA from the gloves. Okay, anyone else? Anyone else? Anyone else?
Yes, sir.
Hmm.
Uh, I have a question like, why is a
Yeah.
ice cold ethanol or isopropanol used during a DNA extraction instead of water, sir?
Actually, we will discuss this in this portion DNA extraction method. But
Mm-hmm.
for your curiosity, I am explaining here. But
Mm-hmm.
DNA is itself uh, we can say hydrophilic in nature. Okay, I'm just
Yes,
checking
sir.
here. है ना हाइड्रोफिलिक होता है because it has negative charge on it. But when you are using water,
Mm-hmm.
okay, it will very difficult for the extract the DNA from the sample. First of all, it will lead to the contamination of the sample. Secondly, you can't extract the sample from the, we can say, sample from the your sample. Okay, like in a DNA extraction method, you will add ethanol or isopropyl alcohol, है ना?
Yes,
So
sir.
in that, these are all are the organic solvents. Okay,
Yes, sir.
whenever you are separating anything, you just need to separate the, we can say, constituents of that particular, we can say, sample. If we are talking about the DNA sample, so DNA, if we are talking about a DNA sample, so DNA sample is not always available as a raw material, raw form. First of all, we have to remove the cell, we have to remove the protein, we have to remove the all other enzymes. Okay,
Yes.
so first of all, you need to break the cell membrane. Then you need to lyse the cell nuclear content from the cell into the solution form. Then where the three layers actually, not two layers, three layers are formed. One is the hydrophobic nature, one is the hydrophilic nature. So according to their, we can say, affinities. Okay, as per their you know about the principle like dissolves like
Yes,
chemistry
sir.
principle होता है ना like
Yes,
dissolves
sir.
like. So accordingly that we have to make the DNA hydrophobic in nature. Okay, हम क्या करते हैं DNA को hydrophobic बनाते हैं, then it will get separated into the organic layer, and other material get mixed into the water. Okay, we have to need to be separate the DNA from the proteins. If we are going to extract the DNA in the water itself, then we will also get the proteins, enzyme, and other RNA, other extracellular material. Okay,
Yes, sir. Thank you, sir.
okay, okay. Anyone else?
Sir,
Hmm.
sir, in the time of fire accidents like that, mass disaster, how we can collect the DNA, sir? Because
Okay,
yeah,
so first of all, we are going to discuss the fire accidental case. Okay,
yes,
so in fire accidental case, if there is severe burning of an individual, okay,
yes, sir.
like you can't identify the different victims. They're
Yes, sir.
totally, we can say, burnt and dead on the spot. Okay,
Yes, sir.
but you can't identify. So for that, first of all, if the body is intact, then you need to transfer that body to the mortuary and for the PM, means postmortem. Then the
Yes, sir.
doctor will collect the sample like femur, sternum bone, or blood sample. If blood sample is not, we can say, destroyed during fire or during burning. Okay,
Okay, sir.
so that doctor will transfer that sample to the police, law enforcement agency, and that police will submit that sample to the lab, forensic
Okay,
lab.
sir.
Okay, after getting the reference sample from their we can say siblings or we can say parents, they will just match the profile. Okay, this is
Okay,
the
sir.
one scenario. This is the simple one.
Okay,
So
sir.
in mass disaster victims, like you all know about there are some like tsunamis or we can
Yes,
say landslides in Kerala. And I think 2 or 3 years ago, na, there
yes,
was a
sir.
landslide. Look, काफी जहाँ landslide हुआ था, and after that, there were a lot of people were we can say buried in the that
हाँ, sir.
Malwa, है ना?
हाँ, sir.
So from that, we need to collect. If the body is in the proper intact form again, then we need to transfer that body to the mortuary and the doctor will do the PM. But
Sir,
the
sorry.
body, if it's in the fragmented form,
Sir, I
hello,
can't hear.
yeah, am I audible?
Yeah, sir. Now I can audible.
Okay, okay. So If the body is in intact form, then you need to transfer whole body to the mortuary and for the postmortem examination.
Okay, sir.
Clear? And if you have the body in a fragmented form,
Okay, sir.
okay, in fragmented form, then first of all you need to see the surroundings.
Okay,
Okay,
sir.
if you have tissue sample, if you have a five tissue sample, or we can say if you have five bones at different places, then you need to collect a whole the samples from that we can say
Okay,
crime
sir.
scene. Okay, you
Ha,
can't
sir.
like assume that five parts, or we can say five samples can be of one person. No,
Okay, sir.
that is a very unethical practice. Okay,
Yes, sir.
you need to collect all the five samples. Okay,
Yes,
and
sir.
you need to like if it is a bone sample, then you can collect in the like a plastic paper, plastic bag, or you can collect into the paper bag. If it's a
Okay.
tissue sample, then you need to collect in the paper bag. Okay, to
Okay,
avoid
sir.
the bacterial growth. Okay, if it is a blood sample, then again you need to collect in the paper bag. Okay,
Okay, sir.
so these are the different scenario. I think you got your answer.
Yes, sir. So in the time of from the fire ashes, we can collect the DNA of that particular
Yeah,
person, sir.
yeah, sure. If you
Okay.
have you will can't skip any part of the we can say crime scene. Okay,
Okay, sir.
if there are ash samples, what is the ash samples are of clothes which the we can say victim
Yes, sir.
wear that time during time of burning or during time of incident. Then it is obvious that the skin sample can be attached to that we
Okay,
can
sir.
say part of the cloth which after that which was burnt. Okay, then you need to collect that sample. I just attended here one burning case in Andaman Nicobar Islands. So
Yes,
in
sir.
that case a person allegedly set on fire a lady.
Okay,
Okay,
sir.
and that lady was 75% we can say burnt.
Yes, sir.
Okay, now is in under the observation. So in
Okay.
that case, when we entered into the house, when we entered into the crime scene,
Okay,
so
sir.
there were a lot of smell of the petroleum products. A lot
Ah,
of
sir.
smell.
Ah,
Okay.
sir.
First scenario explained us that the petroleum product was poured on the lady
Yes,
or in
sir.
the house.
Okay,
Okay.
sir.
Secondly, you can't give your opinion at the crime scene that it will be of petrol, it will be of diesel, it will be of kerosene, it will be of spirit. Okay. The investigative
Okay.
officer will ask you about the we can say specification of that petroleum product, which you have to deny. You have to
Okay.
collect the swab sample from the portion where the no ash samples were there. Okay.
Okay, sir.
And then after that, when I looked into the crime scene, there was a ash samples, there was a
Okay.
lighter, there was a we can say some samples of tissue containing some cloth particles were also there. Then I collected that samples
Okay,
from the
sir.
crime scene in a
Yes, sir.
paper bag. Okay.
Okay, sir.
So and if you're collecting the petroleum product, then you need to collect in a airtight container,
Ah, sir.
airtight glass container.
Yes, sir.
Okay.
Yes, sir.
So this is the scenario. And when you are on a crime scene,
Yes, sir.
everyone, this is for everyone. When you are a crime scene, you have to make your broad thinking. Okay. ऐसा ना हो कि you are just narrating as your investigation. कि हाँ ठीक है हमें sample मिल गए इसको उठा लो. Actually, some persons doing this type of practices.
Okay, sir.
Okay, in my experience, I saw lot of the people which are not of the forensic background. Okay, in some
Yes.
states they are just not forensic background, they are of chemistry background, they are of botany background, they are of biotechnology background. They do not know about the crime scene. Okay, they
Yes,
just
sir.
we can say pack any unwanted evidences from the crime scene, and they just left that important forensic evidence from that spot on that spot.
Okay,
Okay,
sir.
they do not collect. So you need to like have a very broad thinking when you are on the crime scene.
Okay, sir.
Okay, don't worry. I will like a request to the Ayush that one session can be we can say make a workshop on a crime scene investigation and all. Okay,
Yes, sir.
that what are the precautions and what are the we can say steps you need to follow because you are like if you are studying crime scene investigation in your books. Okay, so
Yes, sir.
that is a very totally different scenario from at the real life situation. When you
Yes,
are
sir.
on the crime scene, you just need to like calm. ओके पहले तो काम
Yes.
होना है बिल्कुल शांत होना है आफ्टर यू नीड टू स्टार्ट योर वर्क
Okay,
ओके
sir.
आई
Okay,
थिंक
sir.
इट्स सफिशिएंट एनीवन एल्स
Okay.
Sir, I
Thank you, sir.
have a doubt.
यस
You mentioned that in the forest fire cases or something, we can still collect the samples of ashes, right, of the bodies.
Yeah. Yes.
Yeah, so basically, my doubt is that in normal PCR itself, the DNA will get degenerated around 99 or maximum to 100 degrees Celsius, right? But during in
Hmm.
the
Hmm.
forest fire cases, the body would have been burned for almost 500 to 800 degrees Celsius. So won't the DNA be destroyed or something? So what is the point of
Hmm.
collecting the ashes, or do we still have any other methods of collecting DNA or any other parts where
Yeah.
the DNA will still be alive?
I got your question. So in forest fire or any in burning case, है ना, when there is a total 90 to 100% burning, we can say that tissue is totally burnt, but we are just left with the bones. And bones are itself, we can say, क्या बोलेंगे कि bones are itself as a preservative for the DNA samples. Act as a preservative. Okay, act as a container. So bones can't be, we can say, charred एट अ लो हीट ओके उनको काफी ज्यादा हीट चाहिए होती है बॉन्ड्स को डिस्ट्रॉय करने के लिए यस इफ इन सम इंस्टेंसेस इफ योर डीएनए इज डिस्ट्रॉयड बाय द बर्निंग okay and due to any some fire incident then when you are on a PCR actually you have to before the PCR you need to go with the क्या होता है वो RT-PCR ओके आरटीपीसीआर मैं आपको कल ही बताया था कि आरटीपीसीआर इज रिक्वायर्ड बिकॉज वी नीड टू क्वांटिफाई द अमाउंट ऑफ डीएनए if that amount of DNA is present in a sample, then we can make the profile of the DNA. Okay. Otherwise, we can't make the profile. Means already explained here that कि the degraded DNA can't be analyzed in the DNA forensic lab. Are you getting my point?
Yes, sir. Yes, sir.
Yeah. So basically, we will go to the bone sample. Okay, in fire or fire cases like white fire cases, we will go to the bone sample. If a bone sample are also we can say charred. Okay, because at a we can say point of ignition, we can say at a point of fire, the bones are also degraded, and it will affect our DNA sample. Okay, so we need to quantify. Priorly, we need to quantify the DNA to assure the quantity of the DNA which we required in the PCR. Okay, there are a lot of scenarios, बच्चे. There are a lot of scenarios in forensic lab. इसलिए मैं आपको बोल रहा हूँ कि are you comfortable in Hindi?
Yes, sir.
Okay, so इसलिए मैं आपको बोल रहा हूँ that if you will work in a forensic lab, then you will encounter different type of like in DNA division, you will encounter different type of samples which are in a degraded form in which the DNA sample is not sufficient, then you are not like supposed to be give a final report. You can also there are different actually एक चीज मिस हो गया इसमें I think आगे आएगी पता नहीं मिस हो गया. So there these three type of results are there in the while we are reporting any DNA forensic case. It it can be of inclusion. Okay, it cannot, and we can add a can word, we can add a may word. It may be of inclusion, it can be of exclusion, and it may be inconclusive. So for degraded sample where the DNA profile can't be generated, so we will give the inconclusive, report. Okay, though so at the end line हम क्या लिखेंगे? The results of the detection of the gamer sample we can say are inconclusive because we can't have a sufficient amount of DNA. So that is a limitation, or limitation are always there in every field, है ना?
Yes, sir. Got
Okay,
it.
okay, got it. Okay, okay. Anyone else? So
Thank you,
which
sir.
we are
Sir,
going to conclude a session here. Yes, ask me. Yeah.
sir, so instead of like if DNA is degraded, we can use protein profiling instead, right?
Yes, well, we can use protein profiling, but protein profiling, you need a tissue sample. Yes, you can use also a bone sample, or we can say other sample, but protein profiling has its means we can say उसकी खुद की भी एक limitations होती है protein profiling की. Yes, we can go with the protein profiling. Hello, hello.
Yes, sir.
Yes, and like are you comfortable in Hindi?
Yes, sir.
ओके लाइक आज के सिनेरियो में क्या है कि द डिवीजन बायोलॉजी एंड सीरोलॉजी आर नाउ वी कैन से डिमिनिशिंग। जैसे जैसे डीएनए राइज होएगा ना, एज वी कैन से, क्योंकि डीएनए इज नॉट प्रॉपर्ली वी कैन से एडवांस्ड एट दैट से बिकॉज ऑफ द कॉस्ट। ओके, इसमें काफी ज्यादा पैसा लगता है, काफी ज्यादा पैसा लगता है। सो in biology division we just do the blood grouping, but if we have a dna, if we have a dna, then why we need blood group, है ना? That is simple one line answer is that we just go with the dna, and the biology and serology division are going to diminishing. The not biology because there are so many samples are there like a higher sample, but at the end in forensic science we have a identification, individualization, है ना? ये दो तीन प्रिंसिपल होते हैं ना फोरेंसिक के। फर्स्ट ऑफ ऑल आइडेंटिफिकेशन, सेकंड इज इंडिविजुअलाइजेशन, देन कंक्लूजन। ओके, हेलो एनीवन?
Anyone, if you have any doubt?
So we are going to conclude session here otherwise. Yes. Okay, Priya, please conclude the session if no one have any question.
Fatima, if you have doubt, you can ask.
Sir, am I audible?
Yeah, you are audible. Priya, please just listen to the question.
Okay.
If identical twins have almost identical DNA profile, so how can forensic investigators determine which twin left DNA at a crime scene?
And how it is identified in a real case scenario.
Okay, for identical twins, it is not almost, it is totally same genome. Okay, for identical twins, the DNA of both the siblings, okay, is same. So we can distinguish the sample, or we can say we can individualize the person based on their fingerprints, okay, or other eyewitness evidences. What if for the DNA analysis? What if for the DNA? Okay, the identical twins have the 100% similar genome. Okay, we can't individualize that two twins based on their DNA profile. आपने एक मूवी देखी होगी, देयर आर मूवी, मैं मूवी का नाम भूल गया, एंड दैट इज बच्चे देयर आर अ लॉट ऑफ case studies are there on this scenario. There are two identical twins, and court can't, we can say, identify who is the prime accused or prime suspect. We can say prime, we can say कुछ भी हो सकता है कि कोई भी accused कौन है, but जब वो था, मीन्स एक सिनेरियो होता है
Sir, one thing,
जब
sir.
वो,
Sir,
हेलो,
sir, but yes, sir.
यस,
Sir, but eyewitness cannot be taken as a standalone evidence in courts. Court, no, sir.
eyewitness cannot be taken as a standard evidence, but it can be a corroborative evidence. Okay, so corroborative evidence are
Sir,
those evidence which are
then
supporting evidence. हाँ,
Okay, so then there must be a DNA evidence to support that, sir. If
which DNA evidence are also not acceptable in the court. You know about the some one case study कि DNA reject the, sorry, court reject the DNA report. Supreme Court rejects the DNA report in one case. I just forget that case. Just search and you know कि DNA के ऊपर अभी चल रहा है वर्क, मीन्स इन ऑन द बेसिस वी कैन से लॉ, ओके, कि इट शुड बी, इट मस्ट बी एक्सेप्टेबल इन द कोर्ट, बट डीएनए इटसेल्फ, लाइक एक केस हुआ था, इफ यू हैव हर्ड अबाउट द न्यूज़ फ्रॉम लास्ट 2 or 1 year, कि देयर वाज अ फोरेंसिक साइंटिस्ट has the qualification of MSc botany or also PhD in botany and was working in the DNA division. And the defense lawyer just raised the question on her, I think it was a lady officer, on her qualification. Okay, and then the Supreme Court questions on her qualification that you are a botany student, you have a qualification of botany, then how you can like make a DNA profile on humans? Okay, then DNA, sorry, then Supreme Court rejected that. We can say report of the DNA profile. Okay, there are so many scenarios are there. Okay, and DNA itself is not a we can say acceptable in the court. Huh? It can strong the case. It can strong the case. DNA है ना काफी strong कर सकता है case को. And the final verdict, most of the final verdicts are being given based on their DNA reports by the judge, by the court.
Yes.
Okay, okay. Anyone
Okay,
else?
sir. Thank you.
Thank
Yeah.
you, sir.
Okay, so thank you so much, sir, for today's insightful session and for continuing the discussion on the topic. Thank you to all the participants for joining today's session. We will meet in next session. So have a great evening
So,
ahead. Thank you.
प्रिय जस्ट वन अनाउंसमेंट हियर प्लीज डू योर असाइनमेंट ओके जो हम मैंने आपको एक असाइनमेंट दिया था असाइनमेंट व्हिच आई ऑलरेडी गिवन यू ओके प्लीज मेक सम क्वेश्चंस or we can say conclusion of possibilities okay in that scenario what can be the possibilities okay yes with this Mahalakshmi.
Sir, sorry for the delay, sir. Like, I have a question, sir. Like, I'm pursuing MSc Forensic now. Like,
Yeah,
according to you, like, which evidence requires more kind of research and development for analysis, sir?
which type of sample
Evidence,
I have not
is
huh.
like biological evidence.
So actually all the bodily samples or the biological sample are totally analyzed by a different aspect of the research. है ना? So there are different, we can say, you can add your new approach,
Yes, sir.
okay, based on your research. Like if you want to do a research, like you are asking me question regarding your research topic, है ना?
Yes,
Am I
sir.
right?
If you say so, it will be helpful for me to do so also, sir.
Okay, okay. So yeah, sure. So you can work on your earbags.
Yes,
Okay,
sir.
you can work on I have a lot of ideas, but the limitation for is that you have to like collect the samples, a lot of samples.
Yes,
Okay,
sir.
but that is a little bit we can say difficult. Okay, but you can collect you can work on the earbags. You can work on your जो dandruff होता है I think you are comfortable in Hindi.
No, sir. Only English, sir.
Okay, okay, okay. Like the dandruff in your hair.
Yes,
Okay,
sir.
you can work on that topic. And for individualization, you can work on the population study. For that population study, what you need to some ethical permissions. That is a very vast thing. Okay, but
Yes, sir.
in your masters, you can do on your earbags, hair dandruff, and also on the some we can say you can take the some bodily fluids collectively like semen,
Yes, sir.
blood, sweat, okay, saliva, and accordingly you can made your research synopsis. Okay,
Yes,
but you need to read some research articles before that.
yes, sir. Yes,
Okay,
sir.
then for a sure you will get your research topic.
Yes,
Okay,
sir.
I also did a research on the burnt blood. Okay, because I was reading some research paper. Then I did my research during my masters.
Yes, sir.
Okay, so you can get your answer, but I can help you. You you are from I
Sir,
think
I'm
south.
from Tamil Nadu, sir.
Okay, so Tamil Nadu there is I think coastal area, na? So you can work on the coastal life like. Like you can add on a scenario in the forensic science when anybody when a accused allegedly murdered any person and he just go to the seashore and just put the body into the dump or we can say in the sea and that person that the shoe of that person contains some algal or we can say fungus or we can say some microorganism. Okay,
Yes, sir.
so you can just connect the two links between the crime scene and the suspect and the victim.
Okay,
Okay,
sir.
because after committing the crime, he or she will go to his or her house.
Yes, sir.
Okay, and there we can say that for sure he or she will have his or her clothes or we can say belongings. Okay,
Yes,
connected to
sir.
the crime scene, which can link to the crime scene. Okay,
Yes, sir. Yes, sir.
yeah, yeah.
Sir, also, do we have any test to differentiate peripheral blood and menstrual blood?
but there are a lot of research has been done on this topic. You can just put a title that is the peripheral blood and menstrual blood differentiate. We can say how we can distinguish peripheral and menstrual blood. Okay, you
Yes,
will
sir.
get a lot of research papers on that.
Okay, sir.
Okay, on menstrual blood, the most we can say a lot of study already done.
Yes, sir.
Okay, so I think you will face some difficulty while like getting any such idea from
Yes, sir.
menstrual blood. Okay, but you need to like in the search field you have to select a unique topic. Okay, which help in the social welfare, or we can say which we can say aid the forensic science.
Okay, sir.
है ना?
Yes, sir. Thank you so
And
much,
for
sir.
sure, I can also like give you some topics after
Yes,
some
sir.
time. Okay,
Okay, sir.
okay,
Yes, sir.
okay, yeah.
Thank you, sir.
Okay, everyone. So complete your assignments. We will give you topics in a few minutes or few hours. So thank you so much.
Okay, thank you and good evening. Good night. Good evening.
Thank you.
Thank you, sir.
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 Okay, bye bye, bye bye, bye bye.